Hello, everyone, and welcome back to Kognito Key. Today, we're diving straight into a fascinating and honestly kind of terrifying hidden world that's all around us. We're looking at microbial shapeshifters. These are pathogens that literally alter their entire physical form depending on their environment. It's a biological survival strategy that's just as brilliant as it is dangerous. So let's get into it.
Okay, let's really think about this for a second. What if a totally ordinary, harmless mold, just sitting out there on a dead leaf in your garden, could completely change its shape the second it gets inside your lungs? It sounds like straight up science fiction, right? But it's real. The moment it enters a human body, it transforms its entire physical structure to become a dangerous parasite. It's basically a microscopic Dr. Jekyll and Mr. Hyde scenario. To understand how they pull this off, we've got to look at the term itself, dimorphism. Breaking it down, D means two, and morphic means structure. So a dimorphic fungus is exactly what it sounds like, a fungus with a dual life cycle. It can reversibly shift between a fluffy, filamentous mold phase and a single-celled, round yeast phase. And the trigger for this massive identity crisis? Well, it almost always comes down to the environment. Now, you might be wondering, what exactly drives this transformation? Well, temperature is the primary trigger here. When it's out in nature, in temperatures below 30 degrees Celsius, the fungus is in its mold phase. It's saprophytic, meaning it just peacefully feeds on dead organic matter and reproduces by throwing spores into the wind. But when that exact same fungus enters a host where the temperature hits that 35 to 40 degree sweet spot, like the 37 degrees of the human body, everything changes. It shifts into the yeast phase. It becomes parasitic, feeds off the host, and multiplies super fast through a process called budding. So let's trace this journey. It all starts out in the dirt. The mold forms a mycelial network, and when conditions get a bit rough, it releases spores. These spores detach and basically just lay dormant in the soil until the wind picks them up. Now, if an unsuspecting person breathes in that airborne spore, it travels straight down into their warm lungs. Feeling that 37-degree heat, the spore immediately ditches its mold identity. It germinates right into a yeast cell, starts budding, and quickly multiplies inside the host's tissues. Section 1, famous fungal culprits. Let's meet the actual pathogens doing this. First up on our list is Histoplasma capsulatum. Now, this specific fungus absolutely loves nitrogen-rich environments. You're typically going to find it in soil that's contaminated with bird droppings or chicken manure. When someone inhales it, it goes after the lungs, bones, and skin, causing an acute respiratory disease called histoplasmosis. So if you're ever breathing in dust around old chicken coops or exploring bat caves, this is the shapeshifter you've got to watch out for. Next, we have Coccidioidomycosis. This one prefers hanging out in the soil of low-elevation deserts and causes a pulmonary disease commonly known as valley fever. We're talking chest pain, fever, sore throat, the whole shebang. But here is a really fascinating key fact. While pretty much all dimorphic fungi relied on temperature to trigger their shift, Coccidioidomycosis is the one big exception. It is literally the only non-thermally dimorphic fungus in this entire group. Moving on, we have Blastomyces dermatitidis. This fungus prefers acidic soil, and it's a cosmopolitan pathogen, which means it's found all over the world. The scary thing here is that when inhaled, it doesn't just hang out in the lungs. It can target the skin, bones, kidneys, and even the central nervous system. It causes blastomycosis, bringing on intense symptoms like excessive sweating, severe muscle and joint pain, and just profound lethargy. Let's quickly look at two more really striking examples. Sporothrix shanki lives in decomposed plants, woods, and peat moss. It causes sporotrichosis, which is actually famously known as rose handler's disease because people often catch it simply by getting pricked by a rose thorn. It causes these distinct bumps and nodules on your hands and arms. Alongside that, we've got Paracoccidioides. Found in the soil, this one causes South American blastomycosis, which leads to some pretty painful skin and mucosal ulcers. So the crucial point is, how exactly do we fight back once these fungi have shifted into their parasitic yeast form? Well, diagnosis usually requires cell culturing so doctors can actually see the fungi, alongside serological tests like enzyme immunoassays or chest x-rays to spot the lung damage. When it comes to treatment, medical professionals rely heavily on targeted antifungal agents. Mild cases get treated with azoles, like itraconazole or fluconazole. But for severe, life-threatening cases, doctors have to bring out the heavy artillery, a powerful drug called amphotericin B. And you know, all of this brilliantly highlights just how delicate the balance of our ecosystem really is. Almost all of these pathogens rely strictly on crossing that 37 degrees Celsius threshold to unlock their dangerous yeast phase. Which leaves us with a highly provocative question. As global temperatures continue to rise, completely changing the thermal dynamics of our soils and environments, how might these thermally triggered shapeshifters adapt? Could a warming world actually expand their habitats and increase their spread? It's an absolutely critical question for the future of microbiology.
Thank you so much for joining me today on Kognito Key for this explainer on dimorphic fungi. I really hope this helped unlock a whole new understanding of the microscopic world for you. If you found this information valuable, please make sure to hit that subscribe button and definitely like and share this explainer with anyone else who loves geeking out over the hidden mechanics of biology. Until next time, keep exploring.